Page 168, excitation. The process of excitation begins with the nerve impulse arriving at the neuromuscular junction. The action potential from the motor neuron causes the release of acetylcholine into the synaptic cleft of the neuromuscular junction. Acetylcholine binds to receptors of the motor end plate, producing an end plate potential that leads to depolarization of the muscle cell. The depolarization excitation is conducted down the transverse tubules deep into the muscle fiber. In summary, the excitation process leads to depolarization of the muscle, which results in the release of calcium from the sarcoplasmic reticulum. The big picture of how free calcium results in muscular contraction is summarized on the next section. Contraction. When the action potential reaches the sarcoplasmic reticulum, calcium is released and diffuses into the muscle to bind to a protein called troponin. This is the trigger step in the control of muscular contraction because the regulation of contraction is a function of two regulatory proteins, troponin and tropomyosin, which are located on the actin molecule. Troponin and tropomyosin regulate muscular contraction by controlling the interaction of actin and myosin. To understand how the release of calcium from the sarcoplasmic reticulum interacts with troponin and tropomyosin to control muscular contraction, let's examine the anatomical relationship between actin, troponin, and tropomyosin. Notice that the actin filament is formed from many protein subunits arranged in a double row and twisted. The tropomyosin is a thin molecule that lies in a groove between the double rows of the actin. Note that your troponin is attached directly to the tropomyosin. The arrangement allows troponin and tropomyosin to work together to regulate the attachment of actin and myosin cross bridges. In a relaxed muscle, tropomyosin blocks the active sites on the actin molecule where the myosin cross bridges must attach to produce a contraction. Remember that, the active sites. There's a good question about that. The trigger for contractions to occur is linked to the release of stored calcium from a region of the sarcoplasmic reticulum called the lateral sac, or sometimes called the terminal cistern. In a resting relaxed muscle, the concentration of calcium in the sarcoplasm is very low. However, when a nerve impulse arrives at the neuromuscular junction, travels down the transverse tubules to the sarcoplasmic reticulum and causes the release of calcium, much of this calcium binds to troponin, which causes a position change in tropomyosin, which then activates sites on the actin are uncovered. This permits the binding of myosin cross bridges on the actin molecule. The cross bridges binding initiates the release of energy stored within the myosin and myosin molecules. This produces movement of each cross bridge, resulting in muscle shortening. Attachment of fresh ATP to the myosin cross bridges breaks the myosin cross bridge bound to actin. The enzyme ATPase again hydrolyzes, i.e. breaks down, the ATP attached to the myosin cross bridges and provides the energy necessary for positioning the myosin cross bridges for reattachment to another active site on the actin molecule. The enzyme ATPase again hydrolyzes, breaks down the ATP attached to the myosin cross bridge and provides the energy necessary for positioning the myosin cross bridge for reattachment to another active site on the actin molecule. This contraction cycle can be repeated as long as free calcium is available to bind to troponin, and ATP can be hydrolyzed to provide the energy. Failure of the muscle to maintain homeostasis due to inadequate calcium levels and high levels of hydrogen ions results in impaired cross-bridge movement, and the muscle fiber loses the ability to generate force, muscle fatigue. The cause of exercise-induced muscle fatigue will be introduced in the forthcoming segment. See a closer look 8.1 and associated figures 8.8, 8.9 for a step-by-step description of muscle contraction. Alright, we're going to hold there and look at no, we're going to keep going to muscle relaxation. The signal to stop contraction is the absence of the nerve impulse at the neuromuscular junction. When this occurs, an energy-requiring calcium pump located within the sarcoplasmic reticulum begins to move calcium back into the sarcoplasmic reticulum. This removal of calcium from the troponin causes tropomyosin to move back to cover the binding sites on the actin molecule. The cross-bridge interaction ceases. A closer look 8.1 contains a step-by-step summary of the events that occur during a muscle excitation, contraction, and relaxation. For a detailed discussion of molecular events involved in skeletal muscle contraction, see reference. Time spent learning the microstructure of muscle and events that lead to contraction will pay dividends later, as this material is important to thorough understanding of exercise physiology. In summary, the process of muscular contraction can be best explained by the sliding filament swinging cross-bridge model, which proposes that muscle shortening occurs due to movement of the actin filament over the myosin filament. Excitation contraction coupling refers to the sequence of events in which the nerve impulse action potential depolarizes the muscle fiber, leading to muscle shortening by cross-bridge cycling. The trigger to initiate muscle contraction is the depolarization induced release of calcium from the sarcoplasmic reticulum. Muscular contraction occurs via the binding of the myosin cross-bridges to actin and the repeated cycling of myosin pulling on the actin molecule, resulting in the shortening of muscle fibers. Muscle relaxation occurs when the motor neuron stops exciting the muscle fiber and the calcium is pumped back to the sarcoplasmic reticulum. Then this removal of calcium from the cytosol causes a position change in tropomyosin, which blocks the myosin cross-bridge binding site on the actin molecule. This action results in muscle relaxation. Cool, so we'll hold off right there. That was page 168, 169 from excitation, contraction to muscle relaxation in the summary. See ya.